Health, Psychology, and Human Nature with André Sturesson. Hey friends, hope you're having a great day. Are you following me on Instagram and Facebook? Please go to Health, Psychology, and Human Nature on Instagram or Facebook, or both for that matter, to get the latest episodes, inspiration, and more. Please take a pause and go there. Also, if you like the episode, please share it with a friend, family member, or somebody else who you think might like it. Welcome back, friends, to Health, Psychology, and Human Nature with me, André Sturesson, a science-focused podcast where we explore, learn, and improve our lives together. How does our microbiome affect our health and how long we live? And how does drugs like Metformin and other drugs affect us through the microbiome? In today's conversation, Filipe shares his views on this and a lot more. Filipe Cabreiro, he is the Principal Investigator of Cabreiro Lab, which focus on how the microbiome regulates the effect on drugs and diet on Healthspan and Lifespan. Friends, I really hope you will enjoy today's interesting episode. Filipe, welcome to the show.
Well, thank you.
It's an honor having you on. I think it's going to be a fascinating conversation. I think the stuff that you're doing is quite interesting. So yeah, thank you for coming on.
Well, thank you for the invitation. It's a pleasure to be here. So looking forward to our conversation.
Perfect. We'll see where it will end up. All right. Yeah. So I think there's a lot of interesting stuff that you are doing, and one of the things that you're interested is aging, and also so how to live longer, and also how to stay healthy longer. So I would just would be interesting to know why you became interested in aging and being healthy and that kind of stuff.
An interesting question. I always, sometimes things choose you rather than you make your own choices, right? So it all started when I was still doing my undergrad. It's back in Portugal at the time, and there were 3 students that were going as an Erasmus, internship. And so there were 3 projects. And at the time, we really didn't, we had some interest in science and as an undergrad, but we didn't have a very clear vision of what we wanted to do. So the way we decided to actually choose those topics was literally putting little papers in a bag and take those.
Yeah.
And I ended up in a lab in France. That was studying aging from more from a molecular perspective rather than what I'm doing now, which is more organismal. But there was an interest in the role of antioxidants in aging. So it all started there. And I had a really, really good experience in this lab in Paris. And then they were happy also with how I performed. So then there was the opportunity to do a PhD in the same lab, which I took. And then that's how my interest in aging kind of grew. So it started literally by chance, right?
Interesting.
This was my dream since a kid. But very quickly, I understood that this was one of the most important questions that scientists need to solve. And I got fascinated by its complexity and by the different aspects that the research that is involved actually in trying to understand it that it brings. So that kind of came from luck, but then there was a very clear fit and liking to it, because then that's what I actively chose when I decided to do my postdoc. That I came to the UK, and that's where I really I wanted to continue doing aging research, and that's how I stayed doing aging research. So luck took me here, but then throughout that process, there were active decisions that made me stay, mostly because of the excitement of the area and the beauty of the science itself.
Interesting. It's interesting how different life events strike. Like some things just strike a chord, and then you're kind of just in a new path almost. So interesting.
Absolutely.
So, yeah.
Absolutely. I think luck. I think we dismiss. We tend to dismiss again. I think it's part of our need to control everything, but we dismiss the role that luck plays in our lives. But then, of course, then there's whether something fits you and whether you fit in that same area. And I think there was that. Otherwise, I wouldn't have stayed to do a PhD and then continue as a postdoc and then further expanded it as a principal investigator. But the very first thing was luck.
Interesting. And one of the things that I know that I'm going a little bit further in the future here now. But one of the things that I know that you've been studying and you've been focusing on is the microbiome and longevity and healthspan. So microbiome and how long we live, and also staying healthy for a long while. It just would be interesting to know, like, how important do you believe that the microbiome is for how long we live and for having a healthy life?
So there's different aspects of this question that I'd like to talk about.
For sure.
The first question is how did I go from aging to the microbiome, right? And now, the microbiome is pervasive in most areas of research, and everyone assumes that it's always been like this, right? But it really wasn't. When I actually initially started studying the microbiome in aging, there's another interesting story on this, which was, so I joined as a postdoc, right, in David Jam's lab at UCL in the UK, and my original project was to actually develop combinations of drugs that we could use to improve human aging, right? We use a model organism to do this. But then, as I'm optimizing and testing these compounds and checking whether they really do improve health and lifespan, the way the system that we use, which is a nematode, basically a microscopic worm, constantly lives in the presence of bacteria. And coming from a very different background, as a PhD student, a molecular perspective, I immediately knew that in this a small ecosystem that we were studying, there were several living organisms. There were bacteria, live bacteria, and there was a host, or in this case, the little nematode. And the field at the time, people always assumed that the bacteria was just being there as a source of nutrition, just as diet. But for me, that didn't make sense because there were literally two living organisms. And I was always curious about that. And I said, well, what are the chances that when you are adding now new compounds, new drugs, how much of the interaction between these two living organisms is important for the phenotype, or meaning for the effects that this drug will have on the host and on the nematode. But I remember talking to people at the time, and they were like, "Oh no, that's probably an artifact. Nobody cares about it."
Right? Yeah.
And this is really interesting because at the time nobody cared that much about the microbiome. Nobody really knew. This was early in the 2008, which was not now 12 years later. Yes, there's this huge boom of microbiome research.
Yeah.
But these were questions at the time that were very, very fringe. But I was genuinely interested in that. So I did all the tests that were needed to, dissect these interactions, and then ended up with very concrete proof that the drug of interest that I was working with, that could make really these organisms live very long and be much healthier. Most of those effects were actually dependent on how the drug were interacting with the bacteria. And then I got super excited, and I go talk to my supervisor at the time, and I told him, "Look, David, this is really amazing."
Yeah.
And he turns
Yeah.
to me,
Yeah.
and this is where sometimes you got to be a bit, stick to your guns,
Yeah.
right?
Yeah. Yeah.
And he turns to me and he says, because he knew the field, right? And he's been in this field very, very long time. And he turns and he says, "Well, but that's just a weird artifact. Nobody cares about that. Why don't you just wrap this up as a little story and publish it?" But I knew there was something a lot more exciting there. And I just continued really trying to understand what was going on at the molecular level, etc. Really, what were the things that were changing in this bacteria that could make this host live longer and better. And then when I had a really complex and very clear story, then this was about in 2013, by the time we wanted to publish the work. And at that time, that was the microbiome was starting to explode. And we had a very original take, and we were the first ones actually to show that the interaction between a drug and a microbe could regulate such an important and complex aspect of human life, such as aging. So, of course, this was then published in a very good journal, and then things just took off from there. And then, I never stopped caring and being interested about the microbiome. And now, 12 years after I made this almost initial discovery, we find ourselves with a lot of work on that. We find many other people interested in this field, and a lot of exciting, super exciting research, really demonstrating from all organisms, really going from these tiny microscopic worms to flies, to fish, to mice, even to humans, to show that the microbiome really plays a key role in improving, in mediating the effects of in human longevity.
That is super interesting. But okay, so let's just get down to the basics a little bit. So what you're saying basically is that different drugs can affect the worm differently if it has like a good or a bad microbiome or something like that. Or
Yes. So the microbiome plays a very complex role on regulating host physiology. It can do this in several ways, either through the, the production of certain molecules or that they contain, for example, in their main membranes that could affect the immune system. And of course, the immune system is very important for human aging. It can also regulate and modulate human longevity by the production of other molecules from their own metabolism. So they take things from the environment, like nutrition. They can transform those products, those substrates, into products that then get released in the gut and then travel through the blood to the different organs to actually regulate physiology. So basically, this extra organ, right,
yeah,
is communicating with the internal organs through the production of molecules that can really play with their function. And I think that this is really where the microbiome can be mind blowing, right? We're talking about a huge community of microbes, but it's an organ a bit different than the others in the sense that its cells are very heterogeneous, although now the recent research in other areas do show also that most organs, the cells are also heterogeneous. But what they do is they need to live. These microbes need to live in good harmony and be modulated by what you eat in a daily basis or by the drugs you take in a daily basis, and respond to those cues by producing molecules that they can release into the human body that then that lead to that individual or host to adapt. And then, of course, this has consequences in their, for example, longevity or general health.
right. So it's almost like we have these little factories, or as you say, like another organ that can produce all this beneficial stuff, and these like factories or this organ, it will produce stuff, like it will produce better stuff if we eat in a certain way or if we take some drugs that are good for it.
Absolutely. So it's an organ that doesn't function in complete isolation from the environment, right? Arguably, diet is probably the most important environmental factor that dictates a lot of our physiology. Of course, genetics are important. this this is debatable. Some people that say that genetics are the most important thing. Some people say it's diet. Some people say it's the microbiome. I think my view on this topic, it's really an integrative perspective where you need to take all these things together: the diet, the genetics of each particular individual, and the drugs that they take, so the pharmacological angle, and try to understand how these different components interact and modulate each other in a way that you end up with a final product that can be, of course, very, very complex to understand. But for me, diet really is an incredibly important selective pressure on these microbes, because, as you said, and well, it forces them to produce either good stuff or bad stuff.
Yeah, exactly.
And that's simplifying it in a good way is really that. You have these machines, and I think you nailed it down very well. You have these little machines, these little factories working together, but it's like in a factory. You put something in, you get something out, right? So
exactly.
If you put good stuff in that these microbes can utilize and produce the right type of stuff or molecules, then the consequences of those molecules on us are much more positive. Let's put it this way.
That's super interesting. So I guess what probably a lot of listeners would be interesting to know then, like, what is the good input? Like, what is the good input to get output in these little factories that are good for our aging? What do you think?
So for aging, I can talk more in general terms first.
Sure,
I think the research is moving into it at the moment, and I want to bring an aspect to this conversation, which I think is important, and again, has been to some level not fully appreciated, is again the complexity of our foods, right?
Yeah.
We tend to simplify everything into lipids, carbohydrates, or sugars, and lipids or fats, and protein, right? We simplify things in those three basically macronutrients.
Yeah.
But the reality is each food that we eat is probably composed of thousands of different molecules, right?
Yeah.
And that's what the microbiome doesn't care just about the macronutrients that you eat. They care about everything that you eat, because even, any molecule, if they have the machinery to use it, they will use it, and they probably will transform into something new. And that's something new. If it's a molecule that is in high abundance, or a product from the diet that is in high abundance, they will transform it into something else. And that could be then, of course, good or bad. And we haven't, we're just now actually really tapping into that complexity of foods and how the microbiome can utilize all those different components rather than just this or that. But if we want to there's one area of research that has had a push more than others, is this idea, and there's a lot of evidence for this, is really that fiber is a very good source of energy for the microbiome to utilize and transform into what we know and call short-chain fatty acids, and those are known to exert positive effects on host health. So that is probably one of the cornerstones in terms of this link between diet, the microbiome, and host health. That's now talking about this in the context of aging. The data isn't quite there yet that we can really say that, again, that fiber, microbiome, short chain fatty acids, and aging isn't quite there yet. But there are there's evidence from other organisms that we have identified molecules that the microbiome can produce that can have positive effects. How diet regulates the production of those molecules, we don't fully understand yet, but there's a lot of work going into this at the moment.
Interesting. So fiber seems to be one of or do you think that do you believe that fiber is the most important thing when it comes to having a good microbiome?
That's, in a simplistic way, I would say, most of the evidence points to that.
Yeah.
And it's good, solid evidence. But I don't think definitely that's not the only thing. It's just people tend in science, often people tend to jump on bandwagons, right? Well, the moment that something, everyone says the same thing, because it's easy to, rather than, when you do a discovery, not everything can be new because it's too complicated otherwise. So it's good sometimes to link to the mechanisms that you have, the things you understand, to things that are already there.
Yeah.
And therefore, I think there's an over-representation of the role of fibers and diet and the microbiome in the literature. But that does not mean that that's the most important thing, or that that's the only thing. The reality is we're just opening to new discoveries, new things, and I think in the next 10 years we're going to discover many more nutrients, many more products of those nutrients that are actually important in the regulation of health and also longevity. But yes, in a simplistic way, fibers are known to be an important source of nutrients for the microbiome, and indeed the products of those nutrients or those fibers is a molecule that has positive effects.
Nice, interesting. Like I, I know that this might, I know that there might not be a lot of data for this, but so what do you believe? Like, what do you believe we will find in like 5 or 10 years of different nutrients that will be good?
Again, it's a new field. I think we will potentially be able, the moment we identify some of these positive molecules, right? That could be used as a strategy, right? Even as a therapeutic strategy to improve health. So by directly administering those molecules, right? So synthesizing them chemically and use them, or we can even, just if we know which microbes produce those molecules and which nutrients are needed, we can then do targeted and almost designer diets that could feed your microbiome to make those molecules be produced, which can then, of course, can have the positive effect. But these are early days. We're really creating understanding how the puzzle really works and what leads to what, which microbes are needed, and so on.
So just so I understand, like, so could it be like, for example, so they, in a potential future, like if a person would come in and they would like, okay, I would like a diet that will optimize my microbiome for aging, or perhaps another person would like to optimize their microbiome for, let's say, heart disease, or for exercise, for example, exercise performance or something like that. So that they could like eat, perhaps that we could get these different diets that would help our microbiome to create these beneficial compounds that would be good for certain things.
Absolutely. So I think that's exactly correct. The other, and you can do it the other way around, right? And this is already, there's evidence for this, that certain microbes, for example, can also transform elements from the food, like from meat, to produce molecules that then are detrimental and will lead to, heart issues and heart problems. So you can also do it in another way, which is you can either restrict the diet from those individuals, on certain compounds that, on certain nutritional components that can be detrimental, or you can even remove potentially the microbes that would be involved in transforming those elements from the diet that produce the molecules that are negative. So there's different ways to go around the problem, but the key thing is really to identify these pairs of nutrients, microbes, and their effects on physiology.
Interesting. Okay, so it's both. Okay, so it's both input, but it's also removing some factories or adding some factories.
Absolutely.
Then.
Yes. Yeah. So it's kind of a directed manipulation, basically, of these microbes, and but of course you need to know who's there, what they do, what they prefer, how do they work in a community?
Yeah, exactly.
It's a microscopic level of a society, right?
Yeah.
Where what you give to that society, to that, then the output will depend on, of course, on the individual composition of the elements of that society and their ability to perform certain tasks. And that's that's the same level of thinking as just at the microscopic level inside of that.
Exactly. So it might even be that we need teams of microbiomes. So we need like teams of factories that will only work optimally or work really good if we have all of the different members, like a American football team almost.
Yes, yeah. So that's the other concept. That's interesting you bring that to the table because that's exactly correct. Some people are developing what we call these mock communities of microbes that we know that potentially working together can produce new things that
Okay.
are very, very, almost like in a factory where you have one person building, you in a car factory. One person makes the wheels, one person makes the doors, and at the end you end up with a car, right?
Right,
So
right.
so it's the same concept where you have one microbe that transforms A to
Really?
B, the second microbe will
Okay.
transform B to C, the third microbe will C to D, and then at the end you end up with a very complex molecule, which in this case would be a car, which is quite a complex thing.
Yeah, yeah.
And that very complex molecule has fantastic effects or in regulating human health. So this concept of in-chain communities of microbes is something people are trying to develop more or less successfully, and it's also a very promising avenue.
Wow. Yeah, because one of the problems, as I can imagine, is that the microbiome. I mean, there's so much stuff there, right? It's not like we have one species or whatever. It's you know a lot more
Yeah,
than me about it, but it must be like millions almost, or at least thousands different ones. And like, what does everybody do, and how do they work together? And yeah, must be quite complex.
absolutely. So that's one thing that, as of course, it's part of its beauty, but it's part of its why things also take a bit longer, right?
For
It's
sure.
its complexity. The numbers are beyond our ability to really understand them. But that's why we're currently developing more and more powerful computational tools that allow us, and also the techniques that allow us not only to identify them with uh, very precisely. But now the tools also advancing in the way that not only we know who is there, we know what they're actually doing almost at the individual level. And that's really pushing the field forward for a better understanding of how we can manipulate them, how we can harness this incredible power that the microbiome has. So it's really, I think it's a fascinating area due to those to that inherent complexity for people who like complex things and puzzles and you find you find things there and areas of research that that will never end basically because of this very complex nature, but of course that's a difficulty too.
For sure. Okay, but just go back a little bit about like how if you want to get like a good microbiome. So you told us about the fiber. I've also heard think I can't remember exactly but it was I can't remember exactly who it was, but I watched another scientist who was doing research about the microbiome who mentioned a study where they said basically that having the biggest variety of different vegetable, like basically different good stuff like vegetables, seeds, nuts, fruits, all this kind of stuff, that is also something that seems to be crucial for
Yeah,
having a good microbiome. Um,
yeah, I agree with that statement. uh for not again, if you want to bring this back to at a microscopic level to a society, even though our genomes are what they are and they're very similar, we're all very different and we all have different preferences, right?
yeah, yeah, yeah,
So we're all happy in different ways, and it's the same thing with the microbiome, right? The microbes are related from a genetic point of view just to a degree of variability, and they end up with having again different preferences for different things. And what is important is having a rich microbiome. Yeah, seems to be one of those cornerstones of the field. Is that diversity is usually very strongly associated with positive effects on health. Therefore, the best way to maintain a rich and diverse microbiome is again using a diet that provides you a very rich number of elements, right? When I first mentioned the complexity of diets is exactly that. A very complex diet just massively increases the number of molecules that are needed to maintain an equally complex and diverse microbiome. So the two really go together, right? So if you want to have a complex community of microbes, you need equally a complex and very rich diet. And that's why, diets that are usually based on processed foods, which are very poor nutritionally, they tend to massively reduce the diversity of this microbiome, and usually the consequences of that is poor health. So there is really a lot of evidence really supporting that view, and I would strongly agree with that statement.
Right. If we take the opposite, because I also saw on your lab's webpage that you also mentioned it's not exactly what we're talking about now, but you also mentioned that sugar could inhibit the effects of, I think, the it could affect the microbiome negatively, I think. Like what stuff are bad for the microbiome?
So that's that study tends to be quite quite complex.
Yeah, I know it's another.
I'm going to try not to go into the very fine details of that because it probably would lose a lot of people. But it is true. What we do are really getting to the nitty gritty is there are nutrients that they can completely remodel and individually change how one of these microbes in the microbiome can function. And it completely stops them from producing molecules that are positively associated with health. With that meaning that, indeed, before we were talking that giving them as much nutrients is good to maintain this community, that is correct. But some of these nutrients can, and in the case we identified, were sugars like glucose, could specifically shut down the production of molecules that are positive. So, and that's where things get complicated. How do we then design a diet that, in one hand, maintains richness, and on the other hand, doesn't interfere with the ability of these microbes of producing the positive molecules that they need to, right? So there's maintaining diversity, which, of course, by maintaining the diversity of these microbes, you're more likely to produce more molecules of interest and positive for the host. And this is achieved with a very rich and diverse diet. But of course, within this rich and diverse diet, there are specific nutrients that you probably need to remove or reduce so you don't get the negative out of its interaction with the microbiome. So, and that's really where things get very complex and super hard to work out, basically.
Right.
And this is what the field needs to move forward.
But would you say that perhaps is like, and just thinking about this as a rule of thumb, whatever, like in just broad strokes, what's bad for us is also bad for the microbiome. So kind of if you go from, one side of the spectra, so if you only eat, pizza, hamburgers, all this kind of junk food, and then in the other spectra, it's, eating these clean Whole30, only organic, great diversity of different vegetables and, good wild-caught salmon and that kind of stuff. Would you say that the same thing probably applies to the microbiome?
I think so. It's very simplistic.
Yeah, I know.
But I see the value of doing this. In a simple way, I would say yes, but it's oversimplified.
Yeah, for sure.
Let's put it this way. But indeed, it goes back to the concept. Indeed, that these burgers, these it's how these foods are done, and these and the richness of what they actually contain. But again, don't get me wrong. There's nothing wrong to eat the occasional pizza and the occasional burger. I do that myself, but it's just got to balance.
No, nobody can eat a burger again ever in the life.
Things, right?
Yeah.
So I think it really is about that. If you eat a burger, eat it, with a rich salad on the side. If you eat a pizza, at least in that day, maybe have a breakfast that provides a rich composition of other things that can be also positive. It really is about balance.
Yeah.
So and it's not going too far on one thing, or maybe too far on the other thing. We do know that even people who have very strong vegetarian or vegan diets, they end up sometimes with other deprivations in other nutrients. So it really is about balance, really. And I think everything in moderation is, I know it sounds, again, actually quite simplistic, and it doesn't sound very scientific for all the efforts that we put into research and science, but really moderation tends to be a good rule of thumb to make things work for yourself, for your microbiome, and the combination of the two. So I would stick to that, actually.
Right. Interesting. Well, another point when it comes to microbiome is, adding factories. Now, adding or removing factories. Like if you would add factories, like if you would like to expand your microbiome, there's this trend now also where a lot of people buy these supplements where they eat probiotics. And what's your thought on that?
Yeah, so it's a big field, right? And it's actually a big industry,
Yeah.
more than anything. The science on that is uh, it's it's I don't find it super convincing.
Right.
That's my personal opinion. Uh, but I think there's potential. I think the problem, in my point of view, is that we just jumped too quickly on that,
Yeah.
right? We just didn't spend enough time to fully understand. Going back to our beginning of our conversation, we're dealing with a level of complexity that we don't fully understand yet. We have made some very key important discoveries, but how all these things work together, we don't fully understand. So adding one microbe could well work. Most of these supplements sometimes aren't even live bacteria, so that their ability as a factory is very limited or non-existent. They do sometimes, by having, one thing I said early on, they even if they're dead, sometimes they have things attached to their membranes, right,
Right.
to their bodies. Let's put it this way. That can elicit an immune response, and that could be positive, it could be negative. Sometimes they don't even have a clue. So I think I would be very cautious with this idea of probiotics. But the concept per se, the concept of adding or increasing the abundance of a particular microbe that is known to have positive effects on the host, is a good one. How we make this happen, it's a much more complicated um, thing to achieve, because sometimes you can administer these microbes and most of them don't even, we're talking about a very complex
Yeah.
ecosystem that is in perfect harmony almost. So the moment you get a microbe that is not a natural member, they tend to fight it off almost, and it's very hard sometimes for these microbes to even colonize the gut.
Yeah.
Right. And there's some really, I think, some of the most solid work I've seen in this topic is probably the work from Aaron Alanav from Israel, where he shows that actually in some situations, actually these probiotics can be even more detrimental than positive, and they can even slow down. If you have a very long course of antibiotics and you need to get back to having a normal microbiome, actually having probiotics can slow down that process of recolonization, of reestablishing an harmonious community. So again, it's just there's a big drive behind the economy for that, of course. But the science, I think, actually the good science is very cautious on it, and I think we need to just do a lot more. But conceptually, and I think time will show that this is the way forward. I think right now is just when it works, it's almost more by chance than by design.
Right. Because I also know that you've been doing some studies on metformin. Um, maybe we could just start off talking a little bit on.
So that's our pet drug.
Yeah, maybe we could just start off like just explaining, just very like in a nutshell, just what metformin is and like why it's important in aging research and all this stuff, because it's become quite famous in aging in the aging area.
Yeah, that's so the metformin is our pet drug. That's the drug we started in 2008. That was a drug that I start looking into in the first place. And the reason why I chose metformin already 12 years ago, it was that there was a push for developing compounds that could improve longevity. And there were some studies from Russian scientists actually showing that mice that take this drug, and metformin, I need to say this, is one of the most widely used worldwide anti-diabetic drugs. So people who need to regulate their sugar levels do take metformin. And what they have observed when they gave these drugs to mice was that the mice were actually living longer and they looked healthier. And then triggered an immense amount of studies trying to understand how was this possible and by which mechanisms, basically how was the drug doing this.
Right.
And doing a lot of work, they basically realized that metformin was making the cells of the people who were taking it basically to adapt a state that resembled people that were fasting. So now I need to introduce and say something else. So one of the main concepts, and probably one of the main strategies that is widely accepted as one of the interventions that will improve health, late life health, and potentially longevity, is fasting,
right,
or caloric restriction. So basically, what we found was that, well, what many scientists found was that metformin was indeed mimicking that sort of state of caloric restriction. And the beauty of this was that you could benefit from caloric restriction without actually performing caloric restriction. So caloric restriction works super well in yeast, in worms, in flies, in pretty much any organism, even in monkeys. So and you get all these benefits. You get the late life health. You get the even the longevity associated with it. So people have been trying to find more and more molecules that can basically do this, because for humans, there's a handful of people who can endure going through caloric restriction to gain those benefits. But for our lifestyles, it's really hard to continuously be in that state. And it needs to be done very carefully because it can also be detrimental, right? So therefore, finding this magic bullet where you would say, all right, I can just eat anything I want. I can do, and I just take this pill and I will get the same benefits then of people that are doing this very hard protocol just that they can live healthier and longer, right? So that was kind of the whole thinking behind drugs that could mimic that process. And metformin seemed to be a very good candidate to do that. So that's how I started working with metformin. And I realized that actually a lot of these caloric restrictions or fasting phenotypes that the drug was inducing on the host was actually working through the bacteria. And we kept pushing, we kept trying to understand what was going on and how was this possible. And that's where we discovered that then eventually the drug does alter how the bacteria works and makes them produce molecules that can induce that caloric restriction state.
If we could just pause there for 1 second. So what you're saying is that metformin,
sure.
it didn't add any new, it didn't add any new microbes or any new factories. Instead, it changed them. Like they made them produce different stuff. Is that what you're saying, or?
So yes, that's correct. So that's what our work shows. But our work also in higher mammals and in humans, it shows that people who take metformin, right?
Yeah.
Patients with type 2 diabetes that take metformin will have their microbiome change too.
Okay.
So the drug works in two ways. It works by completely changing the community, so the different types of factories that are available, but then it also changes how these factories actually work. And this is really why I think it's a fascinating drug that has these different ways of working and different ways of impacting the host health. And that's really beautiful work also from from other mammalian sciences, where they can, if you take the microbiome from patients that that are under metformin, and you just take their microbiome and you put them in mice that are not being treated with the drug, you can actually harness the positive effects. So changing that community and their function is sufficient on its own to produce positive effects, and this is really remarkable, I think.
Does the data suggest that metformin is like a good probiotic then? Like it's one thing that actually makes your microbiome better, and yeah.
So I wouldn't call a probiotic I don't want to be pedantic too much.
No, go ahead. Go ahead. It's
a probiotic has to be a living organism, right?
Okay. Sorry.
This would be a targeted. Yeah, it would be basically a drug that you can use to modify the microbiome.
Right.
Yes. It would be a fantastic drug intervention for the microbiome. Yes.
But
what you were saying is correct. There's sorry.
No worries. No, yeah, no worries.
No
worries at all. Okay. So,
but the concept is absolutely
exactly.
Right. It's a fantastic intervention for the microbiome.
So people, but like if people take, let's say that if people would take my metformin for a couple of weeks or a couple of months or something like that, if they were dealing with diabetes or something like that, would the changes in the microbiome persist for a long period of time then, or?
Very, very good question. It's an excellent question. I haven't seen. There's no studies really on that. And most the problem with that is people who get on metformin rarely get out of it, and when they do, they go into other drugs, right? Because metformin eventually, to keep the type 2 diabetes at bay just becomes over time eventually not efficient enough, and you need different drugs. So it's type 2 diabetes it's a disease that you manage. It's not something you completely get out of it. And that adds another element to this, which is something that Nir Barzilai in the US is trying to do. We don't know the effects of this drug in healthy individuals. So it seems to do amazing things for people with type 2 diabetes, but we have barely no evidence of how it works in healthy individuals. And therefore, you're addressing your question, we don't know whether, even if you do almost this pulse chase experiment where you treat an individual for a specific amount of time, whether those are long-lasting changes. From the knowledge that I have about how the microbiome works, I would say it will probably go back to its previous state.
Interesting.
And the reason for that is that the microbiome has an incredible ability to adapt, right? So the moment you remove this constant selective pressure, it's probably just going to revert to its effect. But I don't know. Well, we don't know the long-lasting effects on the microbiome post-treatment. And I think this is a very important question. I would love to see evidence that those are long-lasting changes, right? But usually with drugs, even for the human tissues themselves, right? You correct the defect while you're on the drug. The moment you stop the drug, right?
Yeah.
You go back to the initial disease state, right? So I think I would say that it's the same thing with the microbiome in the context of metformin. But again, unless it does something like a an antibiotic that would completely erase a species from the microbiome that could not go back, I find that it's very likely that he would just rebound.
Okay, interesting. But it's very interesting that you found that. I mean, one of the ways that metformin actually works is because of its effect on the microbiome. And was that like a totally new, like, was that a novel finding?
Yeah, that was absolutely a key finding in that we published in 2013. And others then. So when we published our study first, then there were many other studies in mice and in humans showing that this is actually happening. And that really pushed the field of these drug aspect of the microbiome, because for a very long time we know that microbes respond to antibiotics, but there was barely anything known, to almost nothing, about how it responds to drugs that you take,
Yeah.
basically to treat the human cells. And now we're discovering that these drugs are not innocuous on the gut microbes, and it changes them. It changes the composition or the members of the microbiome, and it changes their function, how they work. And I think that really opened up to an entire new research area, which now is just building more and more and more, and this fantastic work from colleagues all over the world on this topic, which is really an area to keep an eye on, because I think it will probably discover things in the short term that can be applied to humans to really treat disease, to really have a proper impact on human health.
That's super interesting. What makes you say that?
I think it's just based on the information that we already have from the drugs that we have, right? We see that that these drugs have very positive effects, that these drugs are doing something good. We just need to understand it better, and we need to harness that knowledge to make things even better, basically. So that I'm very optimistic that this is really one way to really improve human health. And usually, scientists are a little bit lighthearted sometimes, and they do, when there's a good idea in town, people will follow it. And if it was really such a bad, terrible idea, people will drop it, right?
Yeah.
Because there's such a boom in microbiome research, I think there's a general acceptance that this is a very promising area, right? And funders in the same way. They wouldn't throw you millions of research if they didn't really think that we could really exploit and explore this area in a way that will have an impact, not just in terms of economy, but also, of course, in terms of improving human health. And I think this is really important.
For sure.
So, and again, going back to this in the case of metformin, human data, and this is a fabulous piece of metadata, shows that type 2 diabetic patients who are on metformin are actually living longer than people who are actually healthy, or even compared to controls which are treated with other anti-diabetic drugs. So it's just remarkable that you can be sick, have a disease, being managed by a drug, and this makes you live longer and potentially, I wouldn't say healthier, because health in this context is a bit tricky, but will actually make you survive for longer than people you would consider healthy. And I think it's just remarkable to see such data in humans, right?
Right.
So,
but is it good?
So I think it's just to better understand.
Is there good data on that people who are living with diabetes and taking metformin actually live longer than healthy people?
Yeah. So the data, of course, there's limitations to the data set. Most metadata analyses have limitations, and we need to acknowledge those. But it's there's controls. There's the right controls in those experiments. And this was a data from a Wikipedia study, which was done very, very properly. So I'm confident that does represent possibly what's really, really happening. But again, there's limitations, and we need to be conscious of that. But I think the insights coming from that are very, very exciting, and we need to understand it better and just harness that information, harness that those that understanding to use it in a way that we can make it even better.
For sure. I was thinking about if we would think about the future now, like for a couple of minutes. So let's say that we're in 10 years or 15 years from now. What do you think, like in what direction is the research going? What do you think that we will have found out in 10 or 15 years from now?
That's $1 million question. Humans are terrible. Humans are terrible at predicting the future. So, so what I'm going to say now is probably certainly not going to work. I don't know. I think we will be able to, and we can see already evidence of that. We'll be able to know how to modulate, if a microbiome with very high precision. And I think this is going to be achieved with the ability to test thousands of molecules, with the ability to and have the animal models to test those and prove those. And also the computational tools and the machine learning tools that are now also being used in this context that allows us even to predict molecules computationally in silico that we don't test but could have that effect, and then we can go back and test those molecules. So the landscape or the number of things that we can test now is massively increasing, and I think this will eventually lead to very precise therapies that we can intervene on the microbiome and to then produce the desirable effects that we want.
So interesting. It will be so interesting to see what we will know in 10, 15 years. Yeah, exactly. We'll go back to this. We mentioned before, like we were talking 5 years from now. We'll go back and we'll see what will have happened. But yeah, that sounds really interesting. I mean, if we would be able to really try all this kind of stuff and know more precisely how we can affect these kind of different diagnoses on a more specific level, detailed level, that would be great.
Yeah,
for sure.
I think we're really moving towards this. I think we've spoken about personalized medicine, but this has taken a very genomic, gene approach where we sequenced our genomes, and at that point we thought, now if we know all our genes, we'll be able to do cure all diseases and sort out and design drug therapies that work in a specific way. Then to really realize not far long after that actually we're a lot more complex than we thought. Now we have this community of microbes in the gut that regulates all these different aspects of our diets, of our drugs. So it's not just our genes, really. It's the genes that we have and the genes that we have in our gut in these bacteria. And really looking at the view of these two organisms or meta organism together, is the way that I think we really will be doing solid steps towards personalized medicine.
Interesting. Filipe, it's been great talking to you, really. If somebody would like to know more about the stuff that you've done and the stuff that you do, do you have I know you have your labs page, but do you have any other way to find the things that you do?
So we have a Twitter account, which the handle is Cabreiro Lab. So my last name and lab after. So you can follow us on that. We put updates on on papers that are important and insightful. So that's another way of following our, on what we're doing. There's of course a page of updates in our web labs on the lab webpage, and that's kind of one way to follow us. And of course, if you have specific questions, you can always drop me an email, and I'll always try to find the time to answer.
Okay, sounds great. All right, before we head off, then. What would be your take-home message from today's conversation?
I think people just need to be very aware of what they ingest in a daily basis, and what they do will really have a tremendous impact on how all their lives are going to work out. And I think that's really, and it works out that way because it will affect not only, of course, your own cells, but it will also affect the tiny microbes that live in your gut. And I think that's what I would like to finish on, I guess.
For sure. Filipe, thank you for coming on. It's been a really, really interesting conversation. So thank you. And
pleasure. It's been all mine.
Hope you enjoyed the episode. Friends, I really need your help. I'm trying to get the podcast out there. So I was wondering if you could help me by leaving a positive rating and a review on your Apple device or the podcast player that you're using, as well as subscribing to the podcast. That really helps getting the show more visible on iTunes and other players. And if you don't know how it's done, then YouTube has a lot of great videos, so you can search there. All right, that's it. Take care. This podcast is for general informational purposes only and does not constitute the practice of medicine, including the giving of medical advice. The content of this podcast is not intended to be a substitute for professional medical advice, diagnosis, or treatment.